Hi everyone. Sorry if there's any extra noise in the background from my kiddos. So this is our evolution live stream review. Let me see. Hello, my one person watching me. Let's see. Well, let's see. I will just start talking, and if you guys have questions, let me know. Hi, Cassie. I think it's just you and me right now. Oh, okay, 2 more people. Okay, so just as a reminder, your quiz will cover pages 83 to 94, which is all of evolution, nothing else. So none of the cellular respiration or photosynthesis stuff is on your quiz. Let's see, what else? There is a practice quiz online already, so definitely check that out. Make sure you come with a calculator because you will need to do some calculations. You can probably already guess what kind. No, let's see what else. So let's see, where would I start? Of course, if you have questions, let me know. Otherwise, I will just babble to myself. So let's start with Hardy-Weinberg, because I feel like that's where we started with the semester, so that might be a smart way for us to start. You do need to be able to remember, first of all, that there's sort of a couple different calculations you want to be able to do, right? You want to be able to calculate, if given the information, you would want to be able to calculate the Hardy-Weinberg, allelic and genotype frequencies, right? If you're given the numbers for the different genotypes, then you would calculate the allelic frequencies with that, and the genotype frequencies from that. And remember, all of that would be what you consider observed genotype and allelic frequencies, right? If it gets to the point then where we're asking about Hardy-Weinberg, then now is where you get into the sort of the theoretical realm, and that's where you want to be able to differentiate between my expected versus my observed values. So expected values means that you have you're using the Hardy-Weinberg equations, right? That you have your genotypes, you have your allelic frequencies, your P and your Q, and that you now want to calculate from that what you would expect to get if the population was in Hardy-Weinberg equilibrium. So meaning if you have P and you have Q, and P is the allelic frequency usually of the dominant allele, and Q is the allelic frequency of the recessive allele, then to get the expected frequency of the homozygous dominant organisms, then you do P squared, right? If you wanted to get the genotype frequency of the heterozygotes, it would be 2PQ. And then if it is the expected genotype frequency of the homozygous recessive individuals, then you do Q squared. Chi-squared comes into play kind of in there, but keep in mind chi-squared doesn't have to be for Hardy-Weinberg, right? I mean, we typically for this unit are using it for chi-squared, for Hardy-Weinberg, but it doesn't have to be. Chi-squared is a way for us to compare what we thought we were going to get with what they actually got. And in this case, since we are assuming it's in equilibrium, then that's what we thought we were going to get. So those are our expected values, right? Versus what we got is our observed values. So for chi-squared, I would also give you the equation, right? You'll have observed minus expected squared over expected. And then you would do that for each category. So you kind of also want to see like what do you have, right? Like if you are given only phenotypes, then you would do chi-squared with only the phenotypes. But if you are given genotypes and you can calculate expected genotype frequencies, then you would do it with genotypes. Also keep in mind, a big tip for chi-squared is you always want to do chi-squared with actual numbers of organisms, not frequency. So you don't want to convert anything into a frequency. Like in fact, you would convert it the opposite way, like saying like, oh, well, if I had 100 or 150 or 200, however many, organisms in my population, and that's my actual number of organisms, then I would expect, right? Then if I'm expecting 40% of that, then I would say 40% of my, my population. So 40% of 100 or not, but you wouldn't use 40%. You would use 40% of 100 or 40% of 120 or whatever it was. So just a big tip, because I feel like a lot of times that's where I see people go wrong with the chi-squared. Yeah, for chi-squared, I will give you the degrees of freedom table. You have to know how to calculate degrees of freedom, right? Which is number of categories minus 1. But I will give you that same table that you have in your notes. You do not need to memorize any of that. Let's see. Okay, let's finish off chi-squared. So remember, the idea behind chi-squared is you're trying to see how similar or different are your numbers to what you thought you were going to get, right? So if chi-squared is greater than the critical value, which you look up in the table, then you would say that there is a statistically significant difference between actual and observed values, or sorry, actual and expected values, in which case you're basically saying they're not close enough and they are not the same, which, if we're talking about Hardy-Weinberg, means they are not in equilibrium, right? Whereas if chi-squared is less than your critical value and they are essential, then you're saying that there is not a statistically significant difference between actual and expected values. And there you're basically then you're saying, yes, it is in equilibrium. Hi, are you okay? Okay. You want to say hi, Gina? Okay, go watch your thing. Let's see. Other stuff, obviously, you should know about all the stuff in your lectures. I don't know what questions you have. Let's see. Natural selection, the different types of selection. So, you would want to be able to, understand what disruptive versus directional versus stabilizing selection is. You want to make sure you can understand or you can explain natural selection in general, the idea that, in a population you have, first of all, a lot of offspring, and that they are not all necessarily, that there's variety in that offspring, right? And that they are not all going to be able to survive, and some are going to have advantages over others. So in this case, those who have the advantageous traits would be able to survive, and not only survive, they would reproduce. And if they now can reproduce, then they are going to pass on their genes to the next generation. And as a result, over time, you're going to see the population change to see more and more of those genes, those alleles, I should say, and therefore those traits as well. Let's see. Phylogenetic trees, we talked about those, right? We talked about speciation, so you should know about speciation, being able to read a tree, being able to understand sort of who is more closely related to whom on a tree. Remember that typically we say that the later two things branch off from one another, the more closely related they are. I mean, it's a little too general to say that, but I mean, I think it's sort of a nice overall statement that works. We have our different, you should understand the evolution of eukaryotes from prokaryotes, which we did in a lecture as well. Let's see. Don't know, my 3 people, feel free to ask questions because I'm not sure if I'm covering anything that's actually important to you guys. Let's see. Well, you should know about speciation. I think I mentioned that, right? So being able to know about the different barriers to speciation, or the barriers that exist once species have formed, the idea that a species is defined as a population where they either cannot mate with other groups and make viable fertile offspring, or the fact that they can only mate with one another to form the viable fertile offspring. And that's sort of the key, right? I mean, if they can form offspring but they're not able to survive, which would be viability, or they can't reproduce themselves and they're not considered the same species. Okay. let's see. Questions? My 3 people? Let's see. Maybe I'll take a look at the practice quiz and see what we've got there for you guys and see in there that oh, okay. No, I don't need you to know about the Miller-Urey experiment. I mean, I think I want you to know about it in the context of what it means. I'm not necessarily needing you to, like, quote back what they did, right? The overall idea with the Miller-Urey experiment is that sort of it speaks to how, um, living things would have evolved from those non-living abiotic factors, right? The idea that, you know, Operon had the hypothesis of the reducing atmosphere and the lack of oxygen and the lightning and the whatnot, and then really Miller-Urey just tested that to see if it was even possible, and that it was. So in that sense, you should know that. Um, let's see. Okay, so feel free to type up any other questions, but I'm going to take a quick look at the practice quiz and see if there's anything in there that might, remind you of stuff that you need to know. Let's see. Okay, so let's see. It's gonna pull something up here in a second. Oh, okay. Nope, that's not it. There you go. Oh, uh-oh, is it stuck? There we go. Okay, so conditions for Hardy-Weinberg. Let's see what else. Oh, sorry, I guess there's something that's flipped over there. There's also the gradualism and the punctuated equilibrium, right? The idea of different ways, different theories on the rate of evolution, and just knowing what they are, right? The idea that gradualism is the idea that you have slow and steady change, but a little change at a little bit of time, whereas punctuated equilibrium would be more like starts and stops, like bursts of evolution, and then you know periods of time where there's not much evolution. They now think it's kind of a halfway in between where there might be sort of a little bit of change happening at a time, but then it's not going to be. There also will be times when there's more. Um, okay. Oh, adaptive radiation, right? That's something we talked about in the evolution lecture. The idea that you have like things branching out from a common ancestor. And so, the example I gave you in class was that of the Hawaiian honeycreeper birds, right? The idea that you have all these branches of birds coming out from a common ancestor. And you could also think of that as being related to divergent evolution, the idea that as things become different, they diverge or branch off from one another. Whereas, and those where we have homologous structures, right? Homologous structures are where you have, maybe a similar physical structure to it, even though the actual function is today different, like the cat arm and the whale flipper and the bat wing, things like that. In contrast, we have analogous structures, which is where you have, they don't actually have the same structure, even though they might have a similar function. And so that speaks to the idea of convergent evolution, right? The idea that because their environments maybe have been similar, then they evolved to have similar structures or similar adaptations because they had similar selection pressures. Okay, let's see what else. I look at the practice quiz, I think I also saw things like fitness, right? So I'm just trying to jog my memory too of all the stuff. You need to know sort of the idea of what fitness is, right? Especially in the evolutionary sense. The idea that fitness is a measure of how much you pass down to the next generation, so how much you add to the gene pool. Um, let's see. Let's see what I have else. Oh, you definitely want to know about genetic drift, right? And you want to be able to contrast genetic drift against like natural selection, right? So genetic drift is the idea that there is a change to your population, but it's more due to um, chance rather than somebody being better suited than someone else. And so the two types of genetic drift are founder's effect and bottleneck effect, right? I mean, they both cause you to have this often this much smaller population, and it's a change, obviously, but it's not going to be, due to the fact that somebody's better suited than someone else. Cassie is asking, do we need to know about I would say just know it very generally. There's not too much I need you to know about it. And it's more the AP who indicated they wanted you to have some working knowledge of it, but it's not something that I would say factors heavily into anything. It's just the overall idea that, they think maybe RNA came before DNA. Let's see, what else? Heterozygote advantage, we talked about sexual selection, the idea that, sometimes different genders in certain organisms appear different, and then there might be competition for mates between, one particular gender, etc. The peacocks are a good example of that. Okay, let's take another look at that practice quiz, unless someone's got anything for me. Gene flow, sort of just the big idea, right, of, things, genes moving in or out of a population. And again, remember, once we get back down to it, like at the core, what evolution is the change in allelic and genotype frequencies. Like when you really get down to it, that's how I know the population has evolved. I mean, yes, sometimes I might see like, actual changes in traits and whatnot, but it might other times be just that I see that the population is shifting in one direction or another, towards a particular trait or away from another trait. And that could be because it's being selected for, meaning it's an advantage, or it's being selected against, meaning it's a disadvantage. Let's see, what else can I talk to you guys about? Well, you can see, yeah, I mean, I guess there's not a ton in this particular unit. I mean, population genetics, we kind of already covered with chi-squared. Evolution, we covered a whole ton of stuff. I guess, we could talk about also evidence for evolution. That would be something to know about, right? The idea that there are several different lines of evidence that scientists draw upon that has supported Darwin's theory of evolution, right? Starting with the fossil record and being able to see those sort of changes in organisms over time and being able to sort of connect them to different things, so you can kind of see maybe what, ancestors or such traits that they had. There's also the biochemistry comparisons, which you did in your lab, right? The idea that now we aren't just comparing structures, but we are also comparing DNA, RNA, protein sequences, and the idea being that if they're more similar, then those organisms also evolved, more recently or diverged more recently in terms of evolutionary time. Yes, I can explain that. Okay, so let's go over that. The 3 domains. So when we get to things on classification, it's the idea that once we recognize that organisms have changed, it's also sort of the idea of how do we, divide organisms up, right? How do we how do we essentially categorize them? And so traditionally they did that with the five kingdoms being Monera, protists, plants, animals, and fungi. And those were largely based, I think, on like morphology, right? Like physical structures. And to be honest, at this point, they might end up changing some of that over time because they're finding a lot more evidence in different places that might not always make that quite the right division. But at the moment, it's still what sticks. Um, the three domains is the idea that instead of using those five kingdoms, they used different sorting criteria, essentially. In this case, they looked at rRNA sequences. And when they did, they basically ended up putting things into now three different bins, like three different domains. First, they lumped all the eukaryotes together. So that's the domain eukarya. And then they took that Monera kingdom, and when they looked at the rRNA sequences, it must have been different enough that they essentially separated them into two separate domains, what we call the archaea and the bacteria. Sometimes that's also abbreviated archaebacteria and eubacteria. Okay. Um, and so, again, it's not that it's right or wrong compared to the kingdoms. It's just the idea that now you have a different criteria for sorting things. And so you put them in different bins. That being said, there are some kind of quick ways to figure out which domain something's in. Eukarya is easy, right? If it's a eukarya, it's automatically in the eukarya domain. It's when you get to the bacteria and the archaea that you have to kind of think, okay, well, what else could be different about them? So while again the rRNA sequence is certainly part of that, another part of it is sort of where you find them. The archaea are your more extreme bacteria. You tend to find them in sort of more extreme locations, like really, really hot places, really, really salty, really high pressure, things like that. Whereas the bacteria, or eubacteria as they are sometimes called as well, you would find in sort of more like run of the mill places. It's probably going to be the ones that we, for example, come into contact with a lot more. Um, and so in the end, just want to be able to sort things out and decide which kingdom they belong in, which domain they belong in. And in your notes, we went over sort of like the different characteristics of those. In terms of the endosymbiont hypothesis, that is looking at the idea of, well, how do we see how organisms have um, evolved from unicellular to multicellular? And so when we think about all the things that have to happen in order for a cell to become a eukaryote and be multicellular, um, there are some really key things, right? I mean, assuming now that you already have prokaryotic cells, the endosymbiont hypothesis really kind of speaks to how did all those organelles form? And so you have it in your notes, but it's the idea that first they think that essentially the plasma membrane or cell membrane kind of started to fold in on itself. And when it did that, now you had like different areas, like the Golgi or the nucleus and things like that. So, one part of it is those membranes kind of get it folding in and sort of forming sort of like partitions in the cell. But what's also significant in this case is how you get the organelles, like mitochondria and chloroplasts. Hi, Mr. Chow. Um, so they think essentially what happened is, hello, Mr. Chow. They think essentially what happens is almost like a Pac-Man type action, right? Where you had two different prokaryotic cells. One prokaryotic cell essentially ate the other prokaryotic cell, and the one that got eaten became the mitochondria. And again, the evidence for this is that the mitochondria have their own DNA, like separate from the nuclear DNA that's in the nucleus. And then they think that kind of happened potentially again in certain cells where they already had maybe the mitochondria, but now they ingested yet another prokaryotic. And in this case, it was a photosynthetic prokaryote, and that now became the chloroplast. And again, now the chloroplasts have their own DNA. So that's sort of the evidence to support the endosymbiont hypothesis, which then helps us to understand how we then ended up with, eukaryotic cells which had all these different compartments and organelles and things like that. Let's see. Let's look back at that thing and see what else we got. But feel free to ask questions, like I said, because if it's not helpful, I would want it to be helpful. There's multicellularity, right? The idea that, okay, now once you have a eukaryotic cell, then how do we go from that to a multicellular organism? And in that case, it's the idea that first organisms probably would have started with colonial sort of behavior where they're each individual, but then they kind of share tasks. And then that would be a bridge to eventually having potentially an organism that is made of more than one cell, right? I don't know that there's an exact, idea of how that happened, but we're sort of just talking about major steps. Let's see, what else? I think I talked about it before. Let's see. I guess we could just go off on speciation a little more because I did talk a lot about that. There's a whole lecture on that. So I already defined the species for you before, right? We talked about the idea that they have to be able to produce viable, fertile offspring. So you can also look, don't forget in your notes, you have the prezygotic and postzygotic barriers, like understanding the difference between those and how they work. I mean, the big idea being that, you have these um, things that would sometimes then make it much harder for different species to mate together, right? All the different types of isolation for the prezygotic barriers. I'm going to try and remember them all off the top of my head right now. There's habitat isolation, there's temporal isolation, there's behavioral isolation, there's mechanical isolation. I think there's gametic isolation, right? I mean, basically all these different ways that basically prevent the sperm and egg from even meeting, or at least successfully fertilizing. If they do meet, the idea is that then they wouldn't successfully fertilize to form a new organism. But sometimes that does happen, in which case there could also be post-zygotic barriers. The post-zygotic barriers are things that essentially now, once the um, organism, yes, has, fertilized an egg, is that resulting hybrid organism, most of the time it's either going to have issues with development, or it could be that because of the chromosome mismatch that they cannot successfully produce gametes to make children of their own. So those are all the examples I gave you there, which is like reduced hybrid fertility and reduced hybrid um, what is it? Vigor. I think I forget what the word is in the notes, but the idea that sometimes the hybrids are essentially much weaker, or it could also be that maybe the first generation's okay, but then after that, it's not sustainable, right? So in that sense, you can kind of think of it as it can't possibly be the same species if they can't keep their line going all the way down. In your notes, we also talked about all sorts of different things that would, support speciation, things like opportunity, when there's like there's a lot of opportunity in the environment, maybe because of like mass extinctions like the dinosaurs, and you might now see like more speciation happening or things like that. There's also, we talked about the plants, right? The idea of polyploidy, and the idea that normally we think of polyploidy as not a positive thing, but in this case, if it was able to work out in such a way, sometimes it allows a plant to essentially become reproductively isolated from its neighbors, because of issues in meiosis and whatnot, it can't mate with the other plant right next to it anymore. And usually that's sort of the combination of several steps happening, right? Okay. I don't know what else you want me to talk about. So I will see what I can think of. Let me see. Let me remind you that on the website, you have all the, resources under Unit 4, including the PowerPoints. If it helps you, there are the vocabulary cards. It looks like I don't have the key up, so if you want me to do that, I can paste that as well. Let's see. Is there anything else you guys aren't sure about? You want me to go over? I'm not sure what else. Let's see. I'll take another look at the practice quiz to see if anybody stuff. We talked about phenotypic polymorphism. That's probably a good thing to talk about too. Remember, it's just it's a fancy word for variation, right? And so the idea that is like, where does that variation come from? A lot of it is genetic, but not all. There is the non-heritable variation, but the idea that you can't have change occur in a population, like you're not going to see evolution if you don't have variation in the phenotypes, right? Because if they were all the same, then they would all either get wiped out the same way, or there would just be no variety to be selected for. And I guess that's another thing for us to kind of really quickly think about is the idea of, we talked about at the end of the evolution lecture, but the idea that, remember, natural selection is just that. There's no will behind it. There's no intent. It starts with chance, right? It starts with if by chance there is a mutation or a series of mutations, and now you have all these different organisms with slightly different traits, slightly different variations of that trait, right? That nature selects for the best, the one that survives the best or that does the best there. But that part is not, so the random part comes from the the mutation, but after that it is very much what we call survival of the fittest, right? The idea that the organisms or alleles or phenotypes that have the traits that are best suited to that environment, they will be the ones to survive and reproduce, and then you'll have the population changing more towards that. But on that same note, you kind of remember that at any given moment, the environment could change, right? And if it changes now, who had the advantage before may no longer have the advantage. And so it could very well mean that selection will start heading in an entirely different direction. Okay, let's see. Like I said, feel free to jump in with any questions. There's the idea of sympatric and allopatric speciation, right? The idea that if you have speciation happening when it is due to geographic isolation, then that would be allopatric speciation, whereas sympatric speciation would be a situation where, they're still together, they're still in the same area, but due to mutations or other things like polyploidy, that they are still able to become different enough to be considered different species. Okay, let's see. Let's see. What can I talk to you guys about next? Talked about gene flow, mutations, bottleneck. Yeah, I think we got all that. Okay. I mean, I would say quiz-wise, you definitely want to know about the whole range of, pages 83 to 94, right? That, it's not just one part of it, but you want to kind of understand all of it. Let's see, what else? Well, let's see. Back at the practice quiz again. It doesn't hurt to know about Lamarck and sort of how he wasn't quite right. But again, focus more on Darwin. We talked about Bounder's effect and bottleneck effect already, I think, so probably good there. Yeah. Oh, I guess we didn't talk about vestigial structures, huh? So we can talk about those. Remember the idea behind vestigial structures um, is that they are evidence of evolution, right? They are telling you that in the past there was a common ancestor that may have had this trait. And so even though the organisms no longer had that trait, it suggests that they were, they evolved from something that did. And so it's good evidence for evolution, which again, a lot of times our evidence for evolution comes from um, what is it called? The developmental biology. Oh, Isha. Okay. Um, yeah, I mean, I guess I don't know if I would say yeah, I guess it is memorization, right? I mean, I want you to be able to identify which domain or kingdom an animal would or an organism would belong in, right? I mean, based upon the descriptions. And when you look at the descriptions from your notes, it's not there's not too much going on there. So, I mean, in that sense, hopefully it's not too much to memorize. But yeah, like you should be able to memorize it in the context of then being able to identify which domain and which kingdom an organism would be placed into. Anything else? Let's see. Well, let me take a look at your practice quiz yet again and make sure I shouldn't miss anything. Yeah, I mean, I think we've more or less covered everything. Oh, you're welcome. Let's see. I think I already went over Hardy-Weinberg, but as a reminder, like you should know your Hardy-Weinberg equations, but also know when not to use them, right? I mean, I would say if you have the genotypes and alleles, then you should be able to calculate the allelic and genotype frequencies from that. You should only go towards the theoretical, like the p squared should be q and q squared, if you don't have that information, right? Or if you are talking about something like in theory. Here's what would it be kind of thing. I don't know if that's more confusing though. Yes, you will get your critical values and you'll get your, the basically the same thing that the AP would give you on the AP exam, like I would give you too for this quiz. Okay. Anything else? No. Oh, like you mean like the labs and like the marine mammal lab? No. I mean, especially this time, like the labs, I'm not going to ask you about those specific labs. I mean, we did the labs for two reasons, right? I mean, PTC was to give you practice with all those allelic and genotype frequency calculations. So, in that sense, like, yes, you should be able to do the calculations. You should be able to calculate an allelic or genotype frequency from all different sort of methods, right? Both from the data you got, but also if you are doing it theoretically from, where in the lab, right, we knew the number of non-tasters and you could use that to figure out the P and Q, etc. Like, so there's two different ways to go about it. But like I said, in this case, like for our class, we could totally also just calculate it. And then for the marine mammal lab, that was just to show you sort of the idea of comparing things with biochemistry. But I mean, I don't need you to know specifics about that lab, no. Yeah. Let's see. Anything else? About 5 minutes left. I guess I will just remind you then while I think of other things to tell you. Oh, okay. Why is looking, okay. Well, again, the idea is that, they used to just look at things from the physical standpoint. They would just say, if your structures look similar, like you must have had a common ancestor, right? But now that we know that genetics exists, the assumption is, well, if the DNA is similar, then the RNA would be similar, then the proteins would be similar, right? So it's sort of the same idea that we can apply the same idea of similar structures to similar biochemistry. And so if two species have a more similar protein sequence for the same protein, like in this case, we were looking at hemoglobin, then that would suggest that they branched off from one another later rather than earlier, right? Whereas if we were to see that they were quite different, there were a lot of differences in the protein sequences, especially compared to another organism, then that would suggest the opposite. That would suggest that they, actually diverged from one another quite a long time ago. So, looking at protein sequences in animals is just another way of comparing them. Just in this case, we're not comparing the physical aspect; we're comparing the biochemistry and the protein or the DNA. Um, let's see. Well, yes. I don't know what else people want me to talk about then for the last 5 minutes. Um, yeah. Evidence for evolution. Did we talk about all that? Yeah, I think we did. Or maybe I was halfway through it. Just remember all the different pieces of evidence for evolution. Um, understand about heterozygote advantage and sexual selection. The idea that heterozygote advantage does help us to explain why an otherwise harmful allele would stay in the population. But the idea is that in this case, if having one copy but not two is actually not just okay but beneficial, then you would see it survive better. Especially like for sickle cell anemia in an area with more malaria, then that becomes a distinct advantage. Yes, so endosymbiont theory is just the idea of how did your chloroplasts and mitochondria and other organelles come to be. So think the idea that first there was some sort of infolding of the plasma membrane, and then in order for the mitochondria and the chloroplast to have developed, it essentially became in a Pac-Man type situation where one prokaryote ate another one, and then the one that got eaten instead of and sort of just became sort of an organelle there. And again, the data for that to support that is the idea that you have mitochondrial DNA or chloroplast DNA, like DNA that's in these two organelles that's separate from what's in the nucleus. Let's see. Any last questions? I guess just a quick reminder, since we don't have a ton of people watching, that if for some reason you do think of questions, you can always email them to me tonight and tomorrow night, and I will answer them. Remember to, again, bring a calculator to your quiz. Hi, Ryan. Don't know what else. I think we're nearly at the end here. It doesn't seem like you guys have too many questions, so which I guess is a good thing. But just remember, if you have a question, send it to me via email or come see me during tutorial. Yeah. Let's see. I guess I'll take the last few minutes to remind you of your AP homework since that is coming up. So just remember for your AP homework that you are going to do two different things, right? Number one, you're going to go on to AP Classroom and you are going to do the 23 multiple choice there. It's a set that's already been pre-assigned. And then for the paper version, that's a little different, especially if you talk to someone from last year, it won't be consistent. So just to warn you about that, because we changed the packet a little bit. So you would answer all the multiple choice in the packet in your packet. At least that would be advisable. At least do the first 10 questions. And then on a piece of paper, you'll write your answers to the multiple choice, just the letter like A, B, C, D, E. And then you will answer the one assigned essay question. And remember, everything's based on completion. So you really just for your own benefit want to give yourself practice. Like, yes, you could go find the answers, but that's totally not going to help you. So even if you give it your best shot and you don't write the greatest essay, it's still better practice, right? So that's due not this Monday, but Monday the 10th. And so you're going to turn in on paper the answers to the first 10 multiple choice and the one essay question, which is essay number 7 for AP Week 1. And then online, you're going to go do the online multiple choice by midnight that night. So it gives you a little more time. Okay, so I think we're going to wrap this up then, since it doesn't seem like people have questions. And those of you watching later on, I hope it was helpful too. No problem. All right, have a good night, everyone. And I hope you have some fun tonight, not just studying. Make sure you get some rest. Okay? All right. I will talk to you guys later. Bye-bye. You're welcome. All right. Have a good night, guys.